So, exams are tomorrow, and this is gonna be the best resource we're gonna have. A certain teacher, the GOATS notes. So, when we start, we're gonna cover every single unit. I'm gonna bundle together some of these note packages, but yeah, there's a lot to go through. I'll release them all as separate videos, but to start, there's biochemistry, and IB wants you to understand that there are 5 polymers within the body and 4 monomers, because water is not an organic molecule, and we know this because it does not have carbon atoms. I actually saw a past paper question where they asked if carbon dioxide was organic, but the answer is actually no, because there's no carbon and hydrogen bonding between C and H. So CO2, not organic, and that's something you should know. But yeah, 4 polymers, 4 monomers, proteins, amino acids, carbohydrates, monosaccharides, lipids, fatty acids, and glycerol in a 3:1 ratio. That'll be important for later. And nucleic acids are nucleotides. So, the first part, synthesis and hydrolysis. Synthesis, dehydration, synthesis, or condensation is where you connect 2 of these monomers to create a polymer, such as 2 amino acids to create a polypeptide, or a dipeptide in the case where there's only 2. Some of the suffixes, prefixes, they're kind of iffy. They don't really matter. Like, you could say polypeptide, you could say protein, dipeptide. I think they're interchangeable as long as you know what you're doing. So monomer, what they're saying here is that common within all dehydration, synthesis, and condensation, even if the bond is different at the end, there is water separated by the monomer, and what they're doing is extracting water. By removing water, they create a bond in between, a covalent bond, and you're left with the polymer and a water molecule as the result. Hydrolysis, or digestion, is the reverse of this. You add the water back in to break this bond, creating 2 separate monomers and regenerating the hydroxide and hydrogen atoms. So about proteins, there are 3 groups to a amino acid. There is the amine group, the carboxyl group, and the remainder group, which differentiates the protein. There are 20 different ones, and I believe there were 11 that were essential, meaning that we cannot create them naturally in our the human body, and that we need to retrieve them from food. So proteins are used to build new tissues. They're enzymes. They're building structures. They regenerate your muscles, everything like that. So they're functionally really useful, and they're structurally really important. Like, even your hair and nails are made out of protein. So to create a polymer, so a polypeptide in this case, or dipeptide, you would take the 2 amino acids. The R groups aren't significant yet, but you would take the hydroxide and the hydrogen atom, remove them, and draw a peptide bond where they were removed. So you see how there was a carbon here and the hydroxide group was attached here, and the hydrogen. You'd basically you anneal these 2 with a bond, and this one is called a peptide bond, but I know it's just a covalent bond. In hydrolysis, you would add the water back in, and to show the peptide bond breaking, you would point the water towards the peptide bond like this, and then you would draw this step backwards and get the hydroxide and hydrogen back, and you better have water as a reactant. In the other case, you will want to have water as a product when you're synthesizing them. You can remember this by hydrolysis. Lysis, like the bursting or swelling of a cell. Hydro, like water. So that's how you know it's breaking something. So there are 4 levels for proteins. Primary, simple enough, a long chain. I don't think you really see this in your body. But secondary is where it gets important. The alpha helix shape, come in important when we do nucleic acids, because there's something else that takes this shape, you might know. And beta pleated sheets, which kind of looks like a Z. It's like a 3-dimensional coiling of this. Yeah, so these are the 2 secondary structures. There's not much you can make with them. You have to know that between these amino acids, there are hydrogen bonds, which are connecting them as you coil up and down, which come from the hydroxide groups, should be. Hydrogen bonding to attach between the amino acids. And a tertiary structure is basically a globular shape of these secondary structures, folding in on itself to create like a big blob. these blobs are going to be held together by hydrogen bonds, ionic bonding, which is much stronger, and the retraction and repulsion of these R groups here, because they would be well, some of them are very simple. They might only be one atom, but some of them are polyatomic or even large chains. I believe some are hydrocarbons. So the attraction and repulsion of those R groups is really important in keeping this globular shape. Okay, this is really important. You need to remember some examples of these: spider silk, rubisco, insulin, rhodopsin, immunoglobulin, and they're all globular shaped, like this curled up ball. There are active sites on these structures, allowing them to combine in a lock and key manner. So this is for enzyme function. And lock and key means that there's a specific like match between two things. So they're saying that these are functional proteins, enzymes and hormones, and structural proteins: hair, nail, skin. A quaternary structure is special. A quaternary structure, you might have thought, following the pattern, might be from a tertiary. You would fold this up into the 4th degree, but this is actually 2 or more secondary degree. So you go from 1 to 2 to 3, but then you go from 2 to 4. So it's better to think of it maybe as a Y fork, where 1 leads to 2, but then 2 branches off to 3 and 4. So the example we should know for this is hemoglobin, maybe spelled with an A if you're British, or who knows, IB papers, they might have the A version. Used to transport oxygen and carbon dioxide in our bloodstream. It is should be 4 globin proteins surrounding heme, which is an iron component. And when you have a special group added to a protein, it is called a prosthetic group. Denaturation is the process where protein structure is disrupted. This means that somehow your amino acids are destroyed or somehow irreversibly change shape, and this can happen through heat, like such as when you cook an egg. You know, you can't uncook an egg, obviously. Well, I think there is a way, but leave that to MIT. You can cook an egg with heat. You can use a curling iron, so that'll be really hot, and you can curl your hair, so you'll stay that shape, right? You can use a flattening iron to straighten your hair. Acids, such as when your milk curdles, that's irreversible. And bases, such as when you use a strong base to perm or color your hair. I'm not sure why this, okay. So, onto carbohydrates. Carbohydrates are really important because you need them. You need your blood sugar level at 0.1%. Any more, you have diabetes. Any less, you have hypoglycemia. This is called the homeostatic level. And they're the most readily available source of energy. So, complex carbohydrates and fiber are beneficial to everyday health. And the basic unit is the monosaccharide, as mentioned earlier, such as glucose, C6H12O6. So, it is a hexose sugar. This hex meaning 6. You'll see pentose when you do nucleic acids, but the two IB ones you know is alpha D and beta D glucose. The difference between them is on the first carbon here, their hydroxide and the H atom are swapped. This is a shorthand form using the skeletal structure. If you know how to do it, I think you should do this. It helps save a lot of time. But yeah. On this second carbon here, notice that the hydroxide atom is, hydroxide group is pointing outwards, and the third carbon is pointing inwards. And that's common between these two. It's only flipped for the first carbon here. And this is important when you get to cellulose. So some of the other monosaccharides are galactose, fructose, and then ribose and deoxyribose are special because they're actually nucleic acids, but they use a 5-carbon sugar. The empirical formula for a monosaccharide, this is not for a polysaccharide, is CnH2nOn. Similar to proteins, synthesis of 2 monosaccharides produces a disaccharide with the removal of water. So when you remove water, notice how the hydrogen and hydroxide of these will line up. It doesn't matter which you circle, as long as you circle these ones on the bottom and you take away H, H, and O for H2O. You remove it, and you notice that there's an oxygen left. So what's made here is an ester bond. You might know from chemistry, now that we've done all of OChem. But yeah, Specifically for carbohydrates, this is known as a glycosidic bond. So remember, this is an ester bond, a glycosidic bond. Since we're removing water from the empirical formula of both, we can add them up and then remove 2 H's and 1 O for C12H22O11, which is maltose. Some of the other disaccharides you might want to know, just in case they come up in examples. Sucrose, which is table sugar, comes from glucose and fructose. Fructose is from fruits. Lactose, which is in milk, comes from glucose and galactose. Galactose was from more milk. Some of the other carbohydrates you should know, like definitely know, I've seen them a lot on paper 1. Starch, which is a complex carbohydrate found in plant products. It's the storage form of glucose in plants, and it's branched, but not as branched as glycogen, which is the storage form for animals. It's more compact and easily stored. So it's in the liver, which you'll know from the absorption by the ileum section of the notes in the digestive system. It's stored in the liver and all your muscles. It's good for energy because glucose is a pretty large molecule. The way it's highly branched allows you to store a lot more glucose. Cellulose is not for energy, but is instead for structure. It creates a mesh. I've seen the word mesh used in paper 1. It creates a mesh wall that is very strong, very fibrous, and it's tough, I think, tensile strength when you pull on it. So it's undigestible by humans since we don't have cellulase to break it down, and this is because there's a unique glycosidic bond. Using beta glucose rather than alpha D glucose like everything else here. Beta glucose, since we can see the hydroxide is flipped on the first carbon from the fourth carbon, it actually alternates the direction of the bond up, down, like that. And it forms long chains. It can go up and down as well, so don't get tripped out if you see that. Just be careful because I've seen questions about how cellulose is used to hold in water in plant cell walls, which we don't necessarily think about, but yeah, it's also used to do that. So these are unique, these are still glycosidic bonds, but they just alternate, creating a tough structure that we cannot digest. So for lipids, there are 3 main types of lipids we should know. There's neutral fats, which are made of 3 fatty acids, 1 glycerol. They're just your normal fats and oils. They provide heat insulation for animals. They also do a good job of physical cushioning, protective cushioning, and they provide buoyancy. If you think about whales, dolphins, they're long-term energy storage. If you think about a polar bear who needs to eat up, get fat, so they can hibernate and have a lot of energy. Steroids are lipids with a backbone of 4 fused carbon rings. I think that's just a good definition to know. Cholesterol, testosterone, estrogen, and progesterone, with testosterone, estrogen, and progesterone being the male and female sex hormones. Cholesterol is really important. It's going to show up in atherosclerosis, that process, and it's also going to show up in the cell membrane used to create to keep your fluidity high when your temperature drops. And phospholipid, probably the most important fat, considering that it helps your cell membrane be polar and hydrophilic, hydrophobic. So with that information, you should remember that phospholipids are charged. Phospho for phosphate group, and we know that PO3 2 minus, PO4 3 minus is charged. So back to neutral fats. The basic unit is made up of 3 fatty acids and 1 glycerol. So this is 1 long fatty acid. Oh, by the way, it's called a triglyceride. Sorry. A fatty acid has a carboxylic acid and a long carbon chain. This can be anywhere from, well, I don't know, but it can be very long. You usually don't have to draw this many. You can probably get away with drawing 4 or 5 if they ask you to synthesize a triglyceride, for example. So it's a carboxylic acid. So you can kind of see that this hydroxide group will be used in the synthesis later. This will be the generic formula for a carboxylic acid. You can see CH3 is just this ending here. CH2 is all these repetitions of hydrocarbons here. And carboxylic acid at the end. Don't forget this O is double bonded. And you remember that four bonding sites for carbon, so it must all, this is a fully saturated hydrocarbon. So this is a saturated fatty acid, meaning that every single carbon spot is filled up with hydrogens. It's usually found in animal products, stuff like palm oil, coconut oil, butter, cheese, red meat. And unsaturated fatty acids are much healthier. And the reasoning for this is because they're liquid at body temperature, unlike saturated fatty acids and trans fats, which we'll learn about later, which are actually solid, meaning they clog up your arteries much easier. I believe our teacher mentioned that there have been cases where really bad amounts of fatty acid build up, LDL cholesterol and such. You can physically squeeze it out of arteries and you can see them. You can see the buildup, and it's really bad. So the several double bonds between carbon atoms allow for less hydrogen, so it's less hydrogenated, less saturated, and this simple fact makes the boiling point much lower, the melting point, meaning that it can stay liquid in your body temperature. So these are some safer oils: corn oil, canola oil, olive oil, and fish fats, which you might have heard are really healthy, omega-3 fat. But notice how we know from chem that this must be the first carbon. 1, 2, 3. But if we were to name this alkene, this one would actually be the second carbon. So it doesn't really follow a chem naming scheme, so you might have to be careful with that. But yeah, that's an omega-3 fatty acid. These are cis and trans fats. They're not they're the most unhealthy fats, even though they're unsaturated. It's because of their structure, causing a bunch of London dispersion forces, and their functional group already being a carboxylic. So this is caused by hydrogenation, which you might remember from OChem, where they forcefully fill up the hydrogen to increase the shelf life. So there's fried goods and like stick margarine. Like some margarine is good, like the high quality margarine, no trans fat at all. But if you see this in your products, you should avoid it. And this is glycerol. This is where fatty acids attach to. The hydroxide, they point out this way because oxygen cannot bond. Like hydrogen cannot make two bonds, so hydrogen must be depicted on the outside, I think. So oxygen should be depicted inside. And to synthesize a triglyceride, you're going to have 3 fatty acids and 1 glycerol. You're going to synthesize it 3 times. You're going to draw 3 circles like this. So that means you'll get 3 waters. And all of these are known as simply ester bonds. They're not glycosidic like in carbohydrates. But yeah, they're just ester bonds because there's an oxygen left over. So phospholipids, their shorthand is depicted like this thing, or this Among Us looking thing. They're found in the cell membranes, and one of the fatty acids is instead replaced with a phosphate group. So here, we don't dehydrate synthesize 3, but instead we just have a phosphate group attached, like so. And there is a phosphate OH in the phosphate group biologically, so that will dehydrate synthesize, and I think you'll still result in 3 waters. But notice that the oxygen has a net, well, they're dipoles and they are negative. So this is really important because the head of a phosphate group will be charged, and that's really important for biological functions. Soap, this is crossed out, but basically soaps are fat too, and the charge helps to clean stuff. Nucleic acids are found in cells in the nucleus, and they serve as either genetic material or energy molecules in this form of ATP. But otherwise, DNA, RNA, we'll learn about this later. So the basic unit is a nucleotide, which is a pentose sugar, a nitrogen base, and a phosphate group. So water, final one, inorganic molecule. And I believe we need to know 4 or 5 ways that water is beneficial for the preservation of life. So most importantly, it's polar. Water is polar, meaning that there's a great disparity in the sharing of electrons, meaning it serves as a good universal solvent because of its polar nature. It also has hydrogen bonding, meaning it has a high specific heat capacity because it takes a lot of energy to be able to heat up water by 1 degree Celsius, which is called the high specific heat capacity. And there's also just simple covalent bonding between the hydrogen and oxygen. And the weak attraction means that water has some special properties. So here we go. We need to know these really well. Water is the universal solvent. If you think about it, you drop table salt into a cup of water, it will dissolve and become saline. This is really important for dissolving ionic compounds. It's so strong it can sometimes even dissolve some nonpolar compounds like sugar, soda. The notes are wrong here. Sugar is not polar, but yeah, alcohol as well because of its hydroxide group. And it's crucial because we need potassium, sodium, calcium. We're learning now from the nervous system. Water is also a temperature regulator. The amount of, due to the hydrogen bonding, such as when we sweat, it's good at carrying away heat from our body because of the amount it takes to heat up the water, like to break the bonds and separate water. So water buffers changes in temperature, keeping us at a cool 37 degrees Celsius. Also serves as a lubricant between two surfaces in your synovial fluid in all your bones. And there's also surface tension. It's crossed out, but I'm pretty sure we've learned about this, where some certain insects can land on water, lay eggs on the surface, which is really important. And adhesion and cohesion. In the plant's xylem, water can be pulled up and down to ensure that plants get the water they need. Density, this is the one we didn't learn about. Okay, so acids and bases, this is the probably the most minor point about biochem. So your water creates an equilibrium between hydrogen ions and hydroxide ions. So in pure water, that would be 7, a pH of 7, but our blood pH is slightly basic at 7.4, and this is an important number to know. So this is important because enzymes actually have a optimal range and temperature to function. Like they have a fastest rate at a certain temperature, and some of them only work at a certain temperature. And as we know from earlier, your pH is too high, too low, it'll denature. So your blood pH is maintained at 7.4 once again, and you have buffers in order to prevent changes in pH. So there's substances that can absorb excess in hydrogen or hydroxide atoms. One such is carbonic acid in blood. And you might remember sodium bicarbonate in the digestive system. And yeah, you don't need to know about this. This is chem. So that's it for biochem. The next video should be about cell structures.